Talking here. We're going to be basically, so in your little textbook, you guys have done vocab in chapter 11, so we're going to cover chapter 11. Then we're going to chapter 13 next, because they go together. And I love biotechnology. Who's ever taken biotechnology with Miss Williams? Or taken biotechnology in middle school? A couple of us. All right, so you're going to be familiar with some of this stuff. I teach biotechnology at UNCC in the summers to teachers. I love it, okay? And I've got some good contacts, so I'll make sure that you guys have fun with it. So today is just basically talking about DNA really briefly. I'm going to have you guys draw some stuff and put some stuff in your notebooks. So that's what the people in the front tables, I'll have you guys pass this back. Not yet, but shortly. Alrighty. What is DNA? I'm going to tell you what I feel like is the most important thing to write down. So write it down with me. What is DNA? Basically, you need to be able to tell me that DNA is all your genes. Your genes is, another word for your genes is your genotype, if we're talking about something specific. And your genotype determines your phenotype. That one big phrase is very important. While you're writing that down, I have to get two people to start taking a test. Sitting here? Nay. No. All right, and Isaiah, write down that real fast. I'm going to have you go through the test and put me over there. This is not going to wait for me to do our test. Of course, we. Alrighty, so genotypes and phenotypes are basically determined by your DNA, and you knew that already. You knew the DNA was what your genes were made of. Alright, second thing that's important is this little sequence, because the thing that we are talking about is, it's DNA, it's our genes. The genes are going to code for something, and they code for a trait, they code for a protein, but it doesn't go straight from, here's my gene, ATCG, and I have brown hair. There's a messenger in the middle, because the DNA is lazy. The DNA is where in the cell? Where? The nucleus. Okay, so the DNA is in the nucleus, and it does not leave the nucleus. Well, we have to get proteins to the outside of the nucleus to go. Get messages to the outside of the nucleus, like to the ribosome. Because what is the job of the ribosome? To make proteins. So if the DNA is not going to move, somebody's got to go to the DNA, look at it, look at the message, look at the code, take it out of the nucleus and say, okay, ribosome, I need you to make this. That messenger is called RNA, ribonucleic acid. So since the DNA can't leave, somebody has to go be its little waitress and say, can I take your order? Please tell me what you have, what you need. It's going to go to the chef, the ribosome is going to make it. So there's a messenger there, and that's what this one is, RNA, messenger. The RNA goes into the ribosome and says, please make, using this DNA code, please make this protein. Because this protein is encoded by this person's DNA, and if their DNA says to make it, well, darn, we're going to make it. Okay, so that's why this sequence is very important. So the DNA is equivalent to the genotype or the phenotype? Genotype. So the DNA is more saying, okay, genotype, here's what my DNA says I need to be looking like or acting like. And then the protein, which is, the product, the final product, that's going to be representing your physical appearance, so your phenotype. Because proteins can be, with the way you look, like a characteristic, or it can be like, oh, I'm a PTC paper taster, or I'm not. Because you have to have a protein on your, gene activated on your tongue cells to say, I taste that bitterness or I don't. So it's still all controlled by protein. Okay. All right, and these proteins are going to control a lot of this stuff, particularly enzymes are the most important part. of these proteins that are made. Okay, so enzymes are an important one. Go on the next slide in just a second, okay? All right. You do not have to write all this down, okay? I'm going to tell you, and you're going to write down one thing. 1950s. Who knows somebody who was born or who was alive in the 1950s? Who? Great-grandpa? Grandfathers? Let's see. I think my stepdad was born in like 1954 or something like that. He's old. Okay, he's old. So 1950s, they used to think that proteins were actually controlling they were the ones that were passed on. So like, mom makes her eggs and dad makes his sperm, and they put proteins in their gametes and they pass proteins on because they, like having brown hair was passed on from mom to child, or having hazel eyes are passed on from mom to child or dad to child. So they thought proteins were being passed, but we know what's actually passed. The DNA, right? We know the DNA is. So in the 1950s, they thought proteins were the genetic material because they didn't understand enough about DNA. But we are so far past that. So we've got so many DNA technologies and so much like cool invasive technologies that we can actually see what's passing on. So we know proteins are not it. We now know based off of Mr. Hershey and Miss Chase, or Dr. Hershey and Miss Chase, not Hershey's chocolate, okay? They said, no, DNA is actually a thing that's passed on. So write down what you need to there. But proteins used to be what they thought, and now we know, like, for sure, it's the DNA. Not much right there. I just want to tell you the history of it, because you might hear the scientists, Alfred Hershey and Martha Chase again. Yes? Yeah, I just want you to know that these two people are important because they're the ones who finally said, people, you're wrong! It's not proteins, it's DNA. That's being passed from in the gametes to the offspring. All right, who are some more names that pop in your head that you remember about DNA from like middle school? Yes, Watson and Crick. Those are the next two scientific names or scientist names you should know. Okay, so Hershey and Chase said, all right, no proteins, yes DNA. And then Watson and Crick, I'll go right to them real fast. Lion here. Watson and Crick, these two guys, him and him, I don't remember who's who, they discovered that the DNA looks like this. Can you see this shape? What's that called? Double helix. Okay, so they discovered the double helix. They just, they had some other help too. So I'm going to go back to the part where we're talking about the DNA parts first, and then we'll do the double helix. Okay, so you already know this. We copied this down like 2 weeks ago. If not, if you don't remember it, write it down now. You need to at least draw it though. Draw this right here, something similar. It doesn't have to be beautiful like this. But what I would draw is, all right, so I've got this. Let's do the sugar because I'm going in order. Nucleotides are those, the building blocks of DNA, okay? So the building blocks of DNA look like this, but they have 3 parts. Write down what you need to, but you should be writing something. Drawing something, if you're a writer, you've got to write everything down, you be fast. Right, I would say, here's our phosphate group, and how do I know it's a phosphate group? What's in the middle of it? A P for phosphorus, right. So the P for phosphorus is going to be phosphate group. That's attached to this hex, or a pentagon thing. What did pentagon resemble back in biochemistry? Say. Carbohydrate is sugar, right. So this right here is the sugar, or the carbohydrate. Okay? And in DNA, that D stands for the sugar's name. That sugar's name is deoxyribose. That's a sugar name you need to know. You probably already know it. Write it down if you don't. And then attached to the sugar is this big green thing in this case, but it's different for every type of them or version of them. And those are called the nitrogenous bases or nitrogen bases. So in this big thing, you could just put a big square because it's going to look different on every single one. But this has like a little carbon ring and a nitrogen ring. It's got all kinds of crazy bonds in there. I'm just going to put N base, so nitrogen base. And that's referring to any variety that we have. DNA only has 4 possibilities though. What's the A stand for? Adenine, T, thymine, C, cytosine, G, guanine. So adenine, thymine, guanine, and cytosine. Those are the 4 you need to know. All right, I'll give you a second there if you're processing or drawing. So your drawing probably looks not as technical as that. You probably have a P. You have a. This is gonna be awesome. There we go. That's a pentagon, right? Sugar. Shug. Okay. And then we have nitrogen base. And remember, all your nucleic acids, were they CHO? Or were they CHON? Or were they something else? CHON P, because the CHO version is the carb, the N is from the nitrogen base, and the P is from the phosphate group. We remember, yes. Just say yes and make me happy. Yes.
I remember.
Thanks.
Yes, you do.
Okay. Majority rules. Majority's done. Okay. Listen. Here are the 4 nitrogen bases. You see they are different. Like these are kind of the same and these are kind of the same, but these are totally different. Okay, so that's why I didn't want you to draw like a true structure because they're different for everyone. And you know now know this. Like you could see that and you could tell me, oh yeah, that's a nucleotide. Yes? Question? Or question?
There were like classifications up there.
The purines and pyrimidines? No, that's upper level, and I'm not going to ask you guys to memorize that. All right, structure. Only thing I want you to write on this are DNA. Well, first of all, you know the nucleotides join, right? Well, if you don't, that's one thing that's important. Nucleotides join. So I would write nucleotides join to make DNA strands. Because instead of writing out every single word on the slide, you get to create your own notes. And that's just you summarizing by reading first, listening to what I say, writing down what I say. Okay? Nucleotides join to make DNA strands. And then double helix forms. That's pretty easy, pretty good. And the double helix, and if you want to write some more information, you need to know that it's twisted. How would you draw a double helix? 2 twisty lines. So like, I'm gonna go down a little bit here. So like a twisted ladder, something like that. So here we go. Draw it with me if you want. Draw kind of an S shape, and then you do opposite on this side. And then what are you gonna draw in the middle? Bars. Okay, so this and this, these are 2 different halves of a DNA, of 2 different DNA strands. This first one I drew is going to be a nucleotide, and it's going to base pair with the second one I drew. And it can keep going all the way. And usually people don't draw the space in between. Usually we just draw it like this, and that's fine. But I want you to know that in this line right here, how many nucleotides are attracted to each other? 2. They meet in the middle, right? Okay, so that's what we're going to be doing with our colorful nucleotides here shortly. All right, something else to know is the word anti-parallel. What does parallel mean? Side by side. And the DNA would be going, like from top to bottom and then top to bottom. But to be anti-parallel, it's like this. Okay, so it's top to bottom and then bottom to top. And so it's just flipped. That's it. It's the same thing. It's just one's going upwards and one's going downwards. And we're going to see that in a minute. So I need you to write down the words anti-parallel or the word anti-parallel someplace near where you wrote double helix. Important there. All right, and then you need to know that the 2 gentlemen plus a lady named Rosalind Franklin, she actually helped, but she doesn't normally get credit because she's a lady, and ladies, their contributions to science were not highly valued until about the, 70s, 80s. So it's just one of those things. But anyway, Watson and Crick are credited with discovering the DNA structure as a double helix. Rosalind Franklin. She actually developed a very, very complex technique called X-ray crystallography, and it's very, very hard to read the X-rays, but she could. And she was like, "No, I'm pretty sure it's a double helix." And they were like, "We're not sure, lady. We don't know if we trust your intuition." But she was very smart. Okay, so they said double helix. The only thing that, listen, turn around. The only thing we haven't added about how the structure looks is the DNA backbone. And I'm going to show you that. I think I'll show you in the next slide. I can practice writing right here. So, DNA backbone, don't write this down just yet, is this blue part. You can see the twistiness of the blue part. If I make this bigger, what's embedded in the blue part right here? Sugar, and what's the little circle? Phosphate. You need to write down DNA backbone is, so DNA backbone, because that's a term that we need to be able to understand, equals sugar and phosphate group. I'm going to put P group. So DNA backbone. What is not in the DNA backbone then? Nitrogen bases, where are they?
The ladder, like
The
the
ladder
bar.
rungs, or the rungs that you step on like you were a ladder, or if you were stepping on a ladder. Okay, so that's what they, oops, green mark still. That's one thing that we need to know is what the DNA backbone stands for. Alrighty, now, you know what base pairs are, and you know how they base pair. Probably, but just in case, you know what assuming does. I don't want to assume. You need to know that the adenine base pairs are partners with the thymine, and the cytosine base pairs with the guanine. What is A.T. Allen is in elementary school in the area? Not this area, but like maybe towards A.T. Allen and C.C. Griffin. No, okay. That's how I've always remembered it. A.T. Allen is an elementary school in Cabarrus County, and C.C. Griffin, so C. Griffin, is our middle school, but it's okay. All right, so you'll see that I put a dash between these. That dash is intentional. Please write what that dash means. That dash means hydrogen bonds, okay? So what happens is the adenine thing and the thymine nitrogen base, they have this attraction between them, and that attraction is called a hydrogen bond. So if I ask you, what holds the two base pairs together, you would say hydrogen bonds. Yes, Ty?
You know how you like trying to find something for a number?
Mm-hmm.
Like when that is the C and G, all you have to do is draw one line up to get from the C to G.
Yeah, those letters look eerily similar. Yeah, you're right. And those, yeah, just remember those look really similar. And those are all straight lines. True. I like it. I like it. Okay. And then the other terms that we're going to be using a lot are complementary strand and template strand. So complementary. What does it mean for something to be complementary? Goes along with it. Like you complete me. You are complementary to me. Okay. So the template strand is going to be one strand, and the other one is going to be the template. The template strand is going to be usually the first strand, and the complementary strand is going to be the other strand. So if I had a complementary strand, I'll go right back there. Where's my? There we go. If I have a template strand like this, write down my template strand A, T, C, G, G, C, T, A. Write that down, and write template strand above it. What would be, I'm going to write this one in green, what would be my complementary strand to this one? T, A, G, C, C, G, A, and T. Right, so that's pretty easy. It's just if I say write the complementary strand, you're like, what the heck is she talking about? I'm talking about base pair. That's it. So the first one in blue is the template strand, the second one in green is the fill in blank is the complementary strand. All right. Now, as you're writing that down still, there are some colorful cards on your desk. Those of you in the front row, please pass back. But hold on, everyone's going to get 2 of each color. Okay? Pass back what you need to, but everyone gets 2 of each color. So you should end up with 8 little cards. Pass those back for me. Pass those back for me. Make sure you guys get some. Okay? All right. All right. You're going to be putting this in your notebook or on notebook paper or whatever. I'm going to do that.
So confused.
Just take two of each. One each person. All right, so I want you to. Okay, I'll get you another one. Hold on.
Yeah, I didn't know.
You need one more what?
One more yellow.
All right. If you're missing some, I will get them to you in just a moment. Austin and Chandler are passing out scissors.
We do it once each row.
If there's enough for everybody, try to keep it enough for everybody. If you have your own scissors, pull your own scissors out. I want you to make a DNA template strand and then make the complementary strand. And I will tell you what I want your template strand to be. You will have to cut out only you will only have to cut the part of the DNA that needs to fit together. This part right here. You do not have to cut out the whole shebang. Just leave it in the bucket. You don't have to cut out all of this. You can just make like a little wavy cut like this. The only cut that definitely needs to be a true, good, on-the-line cut is the cut that's going to allow for the base pairs to fit together. Now, look up here. I see your eyes. Let me see your eyes. Why is this upside down? What's the word? Anti-parallel. Right. Okay, it's anti-parallel. Here's the sequence of the DNA template strand, and it will need to be in this order. A, C, G, T. And your job is to make that one just go down the paper, and then make the complementary. I'm saying less than like 8 minutes. In this, yeah, go in this order, and then do your complementary. Does everybody have scissors? Okay, I'll get you another blue. All right. If you are cutting your things apart, be quiet. So I'm going to explain it one more time just in case somebody missed it. You have 2 of each color. You are going to cut really only the part that needs to bond together, the part of the A that needs to be matching the part of the T. And you can make a little wavy cut around it. You don't have to cut every nook and cranny of this big, huge nucleotide. Mainly that part right there. And then you're going to make a straight line down your paper in the order of A, C, G, T. This is called the template strand. TS for template strand. You're going to make its base pairs, and I'm calling that the complementary strand. And we noted that the T was upside down because of the anti-parallel nature of the DNA. All right, raise your hand if you're missing a color. Missing a particular DNA nucleotide. What else is back there? I'm gonna get you guys some tape.
So, just the ones that we connected up next, like, they need to be all on the same.
Because what you're going to be doing is, I have tape and glue. I have to find my glue sticks though. I don't know where they are.
This is how we do it, right? I'm not like cutting it wrong.
That's fine. That's beautiful.
So we're not writing.
No, it's you are making your template strand, yeah. And then you just have to line them up and make sure that they bond together. Make your DNA backbone. What's my next one? C. Does the C have a rounded shape? I'm gonna write this upside down though, because it's upside down. This is looking real good. Okay. You scaled up writing upside down. My glue is. Does anybody else have glue or tape by chance?
I think I do.
Then pull it up you and your quadrant over there. Quad.
Oh, I have it.
Now, are you going in the order given?
Oh, I haven't done that yet.
Oh, you just
I
need
get
to.
on that.
Here's tape for your whole area here.
I just did it. I just laughed at kids who talk. Someone already paid cheap.
Oh my gosh, tape. Wait, not you. Yeah, this tape is really, killing me slowly.
No.
Standard is black. Just a plain black.
Or have maintain your morals and ethics. So, or maintain your morals and ethics.
No, it's fun working with like the coaches because a lot of them.
Or just referee when it's all referee. You can get like.
Guys, there's trash cans around the room you guys can see. Make sure you throw your scraps away. Oh, seated. Alright, you guys should be taking this down in your notebook. I have one glue stick. I'm looking for more. Somebody has saved them from me.
I get paid $7.40 an hour.
It doesn't matter, just whatever they put in.
I'm about to be
Every sound.
Maybe you just stuck with that.
Yeah, I'll do all of that red card.
Anybody a red card in here?
Bracket, you actually have
I will pull out a blue card. You know what that means? You out the league.
that's a red card.
So they're out of the league. No, it's not. Red card is a game.
That's your goal, red card is then playing soccer and
I like when people are like so far away, but they're still in the conversation. Yeah.
So I got mine are like at 14 for the last time. Alright guys, I'm gonna say 5 more minutes. 5 more minutes and that's it. Get it taped down. Oh, thanks, Lauren. You're awesome. There's like 5 tapes down here. Go steal some. Hey, I didn't make it. Oh, look at him.
That doesn't put you when you're on the couch. That's Ripple.
Alright, 4 more minutes, guys. Get it taped down.
I will not work at
I get $5 a game. I do it.
Comfort Mills or
Yeah, I get paid $7. If he doesn't, I see my Twitter. Sell drugs.
Blue intake. Blue intake. Raise your hand. Raise your
Hey, what do you mean?
You're not even doing
I'm in a restaurant, walking up and down the field. You don't see me like what is your
the only thing that hates about those schools is that it's supposed to give back to parents. I had a customer last
I'm getting paid overtime. Yeah, I'll be the one that gets my way off.
Actually, wait, I think they said that.
Wait, but you didn't ask me. I'll be the referee.
I think I was talking to you and you guys like don't have your students or whatever.
One more. And love.
Austin. I worked at a bakery. No, I worked at Coldstone. Coldstone. I got free ice cream and then I got sick of it and I've never eaten it since.
I've never eaten ice cream.
Not Coldstone ice cream. It's not ice cream. It's like super ice cream, but it made me sick.
When'd you get your first job?
My mom wouldn't let, she wouldn't let me work during the school year, so I got it the summer of my sophomore year.
Over half, like,
Yeah,
hours is like,
Lady trying to get off.
Okay. Where did I put my notepad? Alright. There's glue. Get it done. Alright. You have 30 seconds to clean up your desk, done or not. We are moving on. We have 2 more slides of notes. 30, 29, 28. Clean up your desk. Throw trash away.
Hey, who's 16 in here?
Guys, it doesn't matter. Finish this up. We're going to start next again. Please do not make it up.
What about it?
Does anybody else need to get space? Alright. Thank you. Take a big strip of it. Take down the whole thing. Alrighty guys, be quiet. 5, 4, 3, 2, 1. Turn around. Go ahead and write down the steps of DNA replication. We have 2 slides left. After that, we are doing the lab. Your templates will be used again later, so do not lose them. That's why I said put them in your notebooks. You could not lose them. Hopefully, we don't lose them. Alright, I'm not going to stay here for long, so if you are writing, thank you very much. If you are still cutting and taping, you have about 20 seconds to put your stuff down and start writing. I know that you guys don't want to lose your templates, but that's why I said do this in a timely manner. DNA replication. I'm going to be quiet, let you guys write down every bit of this, because this is probably the most technical thing we're going to talk about in the first part of this unit. Oh, I got it. Turn around and write what's on the board.
oh, yeah, that was a bad one.
Is this the worksheet that you were doing?
Writing down everything on the board. Thank you. Sit and look in your papers, not writing. Okay, stop doing what you're doing, man. Write down on the board. If we can't start the lab by 10 o'clock, then we don't do the lab. So, we got like 5 minutes, 10 minutes. That clock is good. I'm gonna scroll down. Alrighty, let's go back to the top. So DNA replication is that's something we talked about way back in mitosis. What phase? I'm sorry, in the cell cycle. What phase in the cell cycle did we talk about DNA replication happening? I hear one correct answer and one incorrect answer. Anybody else? Interphase. Yes, interphase is when this happens because we have to make sure it goes through G1 where it grows, S, which meant synthesis, DNA synthesis is DNA replication, same thing. Then G2, that's the cell cycle with the second growth phase. Then it goes through mitosis, then it goes through cytokinesis. Okay, so that's one thing to remember. We already talked about this, but now we're getting to the gruesome details of DNA replication. Okay, so before the cell divides, it has to be copied or replicated. This is called replication. No doubt, right? Copy, replication, the same thing. All right, step 1. I have abbreviated this on the board. The DNA, here, show me double helix. Double helix. All right, this way. I can't do that way. Double helix. All right, your double helix has to uncoil and unzip. All right, so we have one strand of DNA and another strand of DNA. All right, so double helix has to uncoil and unzip. So unzipping means the DNA nucleotides are no longer attracted by those bonds. What were those bonds called? Hydrogen bonds. Okay, so those hydrogen bonds have been broken. All right, so uncoil, unzip. Then we have to send in some free nucleotides like A's, T's, C's, and G's, and they will go and find their base pair partners, and they will base pair together. So step 2, nucleotides base pair. They're just going to go find them and replicate. And then step 3, there's a proofreading enzyme called DNA polymerase. It has to proofread because, this stuff's happening in nanoseconds. The A's and the T's and the C's and G's are flying everywhere, and they're base pairing with those 2 individual strands. What if they make a mistake? What have we just done to your DNA? There's an M word for that. Mutated it. Okay, we have mutated it. And that can be problematic because a lot of times mutations are bad, sometimes mutations are okay, sometimes they're neutral. Okay, so mutations can be bad. They can mess up your genotype and then your phenotype. So we have a proofreading system that spell checks the DNA. It's going to make sure your DNA is happy and good, just like it was supposed to be. Then the 2 new strands retwine together, recoil together, and come back together. So rezip or recoil is another word to say that. So I'm going to show you guys a video that shows this exactly. All right, this is one of those educational videos.
In order to replicate, the 2 nucleotide strands of a DNA molecule separate at their base pairs. Each strand then builds its opposite strand by base pairing with free nucleotides. For example, if one nucleotide on a strand has thymine as a base, the free nucleotide bonded to it would be adenine. Each new replicated DNA molecule has one nucleotide strand from the original DNA molecule and one nucleotide strand that has been newly synthesized from free nucleotides in the cell by base pairing with free nucleotides. For example,
Sorry. Okay, this right here was the original strand, and then these are coming in and base pairing. What are those things forming between the C and the G? Hydrogen bonds. Good.
if one nucleotide on a strand has thymine as a base, the free nucleotide bonded to it would be adenine. Each new replicated DNA molecule has one nucleotide strand from the original DNA molecule and one nucleotide strand that has been newly synthesized from free nucleotides in the cell.
Okay, so that means every DNA strand is one of the original double helix strands, and then a new one. That has a term. Was that in your vocabulary?
Yes.
What was it called? Was that semi-conservative replication? Semi-conservative replication. That term needs to be written down in your notes someplace, por favor. Semi-conservative replication. Which means, semi means like half. Okay, like a semicircle? Halfway conserved. So, in this case, in my example, this strand here was the green one, and then this was the green one. And when it goes through and replicates, and it splits and unzips right here, one of these strands of the new DNA is going to be the green from the original, and one of these of the new DNA is going to be from the original, and the other one is going to be from those free base pairs coming and bonding and making a totally brand new, spanking new strand. So this is going to be a new one that's going to be embedded with the old one. And that's why it's called semi-conservative, because each of these are semi-similar to the very, very beginning. Okay. Take a stretch break for a second. Stand up. Stand up. Stand up. Stand up. And I have another video for you. Everybody stand up. Everybody stand up. Get off your butts. Because you're going to stand up and do this one with me. Okay. Get ready? This is a biological supply company. Put on my desk. Come back to the front. I didn't say like go to the back and chill by yourselves. I said stand up. That's it. Okay, this is the biological supply company that made this song, and it's hilarious. Okay, you have to do this with me.
Hey, look at our boy now. Randolph. Now, I remember somebody that wasn't too different from this not too long ago.
Show me your body way, let me live you with all my enzyme. Trying to build a new strand, I said enzyme. Cause I know that you can't, I said enzyme. These letters are so champagne, all we need to get started. It's just a little bit of GTC. Just a little bit of GTC.
scientists out there doing PCR, Bio-Rad salutes you with the all-new line of self-fast scanners.
So this is the commercial.
PCR.
Okay. That is a commercial for Bio-Rad, which is a biological. You can go back to your seat now. Thank you. That's a commercial that they're trying to sell a particular. I'll wait on you guys. Turn around. They're trying to sell a particular enzyme that helps speed up chemical or DNA replication that they can do in the lab, not like in the body, but in the lab. Because a lot of times, like who's watched CSI? Okay, you go to the CSI scene, there's a drop of blood. Okay, well you want to see who it is, right? You got to figure out whose blood it is. That blood does not have a ton of DNA in it. So if they're going to take that sample of blood from the crime scene and they want to see whose it is, just in case they make a mistake on the first go with their testing, I mean, what if they make a mistake? All the DNA is gone. They're screwed, right? They're not going to figure out who the culprit of the crime is. So what they do is they take the blood, they take the DNA out of it, and they put it in this thing called PCR, polymerase chain reaction. And they replicate it like 1,000,000 times. Okay, 1,000,000 times. So they've got all kinds of DNA to do tests with to see who the criminal is. They can say, okay, that lab, you guys are going to do this test, we're going to do this test, and like 100 labs later, you're going to do this test. And they'll never run out of that person's DNA, because if they run out, they've lost the criminal. So that's why they have to make this particular enzyme to speed up that reaction, because you can imagine it would take forever to do that. All right, I think we have enough time to do our lab, but you need to impress me with your ability to follow instructions and complete the analysis, okay? We are going to extract DNA. Normally I used to extract DNA from your own cheek cells, but it's really small because we only have diploid cells, so how many copies of DNA? 2 copies, one of each chromosome. Other things like vegetables usually are octoploids, 8 copies of their DNA. So it comes in, it's really visible when you see the octoploid because it's not an octopus, okay, it's an octoploid, because it's got 8 copies, so it's very, very visible. So we're going to use strawberries and we're going to extract the DNA out of a strawberry. Who's ever done something like this? A couple of us have. Okay, you're going to be the pros, you're going to be the leaders of your lab group thing, because sometimes it's very technical on how it rolls, okay? You need a piece of paper out? Get a piece of paper out now. Papel. Finish checking vocab, I'm working on it, leave it on your desk. Okay, I'm going to explain this as you get your paper out. Look up here. I want to point out one step mainly. I already have the DNA, I already have the strawberries at your desk. I already have everything at your desk. There's one part that you have to be very careful with, and it's whenever you add ethanol, which is alcohol, to the solution. So these are the instructions. They're at your lab stations. You're going to take a strawberry in a bag, and you're going to smush it for about 2 minutes. I'm going to wait for people to give me their full attention. All right, you're going to smush it. Then you're going to put some DNA extraction buffer. It's labeled in your lab benches already. You're going to put that in there. You're going to smush it some more. So it's going to extract the DNA out of strawberry. Then you're going to pour it through a coffee or through a funnel with some cheesecloth. I've already got the cheesecloth cut to the size you need. It's going to take a little while to drip. While it's dripping through, you're going to do the data questions because it's based off the pre-lab reading. You don't need data to answer those questions at that point. So while you're waiting, don't sit there and chill and look pretty. I know you guys are good at it, but you're going to do those questions. Okay? Whenever you get to the part where you have the DNA already in the test tube, you need to isolate the DNA from the stuff, from the liquid. That requires ice cold ethanol. When you are ready, I will get it straight from the freezer. I cannot pull it out before because then it's not ice cold anymore. It'll get to room temperature. So at this point, step 6, you have to read all the way through because what you have to do is you turn your test tube kind of to the side and you let it drop down the side because if you put it right in, it drops and it goes through the middle of the solution. And then it's mixing together. DNA, it won't extract. It won't work. Okay, so you have to read that part very carefully. Do not mix the 2 layers is the main point here. There's some other steps after that, but it's all pretty straightforward. If the DNA is beautiful and you want to keep it, I will find a container and let you keep it. Okay? All right, your lab groups are, get ready. All right, lab group 1 through 10, you guys can see your name. I will say, if you did not do your vocabulary, you are going to sit down and start your vocabulary, finish it, and then you go to the lab, okay? So, thank you very much. See you in the lab in 1 minute. Go. Reagan, it is. I added it. It's underneath. Here. Reagan, you see? It's underneath there. You're group 10. You're about to put this on my desk. Lizzie, I'll talk to you in a second. I need to get my class. I need your chapter 11. Chandler, will you finish your vocab? Go ahead and start reading. Chapter 11. You doing vocab? Okay. Lizzie, one second. All right, guys. Read the instructions and get started.
Speech.
Read the instructions. Lab groups, listen, Cycle 321. I need somebody to grab goggles for your group. I'm sorry, I forgot. One person grab goggles for your group. Can I tell you, you have to go on the vocab. Okay. Chapter Okay, I trust you to get this done. You have about 4 minutes to get the entire lab done. 20 minutes. Problem time?
Yeah.
Problem time?
I'm my best friend.
You can do the long one.